Price, the founder and CEO of Academia.edu, and as always, just your friendly host, here to guide you through some of the most exciting, sometimes mind-boggling academic research out there. Today we're diving into a recent paper by Karim Hanid, which has gotten a lot of folks in immunology and cancer research talking. Its title, well, it's a bit of a mouthful, so bear with me. Targeting the root of T-cell exhaustion, the Protectoric StressResponse, as a fundamental mechanism of immunotherapy resistance and a Nobel-caliber breakthrough. Yeah, it's a big one. And just to be clear up front, I'm neither endorsing nor critiquing the findings here. My aim is just to break down what Hanid and the team have found so you can reflect on it and maybe even chat about it over coffee later today. So, the core of the paper, what's the question exactly? It's this: why do so many cancer patients, especially those getting the most advanced, celebrated immunotherapies, fail to respond in a lasting way? Immunotherapy has sort of revolutionized cancer treatment lately, right? Yet, more than half of those battling solid tumors still aren't seeing the durable, life-saving results we all hoped for. That's a huge deal. If you or someone you care about has ever faced cancer, you know just how frustrating it is when you're told, well, the best science out there just doesn't work for everyone. This is where Karim Hanid zooms in, asking, is there a deeper sort of cellular root cause behind why so many patients become resistant to immunotherapy? And the answer, well, in this paper, it circles around this newly synthesized idea called the Protectoric StressResponse, TexPSR for short. So TexPSR, it's a new model, a way of explaining why T cells, these immune superheroes, burn out and become what scientists call exhausted in the heat of battling cancer. Let's back up a second. T-cell exhaustion, that's the big villain here. It's basically when T cells, instead of killing cancer cells, go into a kind of funk, losing their powers one by one until even the most sophisticated treatments can't wake them up. So in its work, it's saying, hang on, this isn't just about immune breaks. This exhaustion is driven by the T cells' inside world. They're protein-making, protein-folding, stress-managing machinery. If you're picturing stressed-out little T cells with papers flying everywhere in their cellular office, well, it's not entirely wrong. According to the research, chronic exposure to tumor antigens, constantly being triggered to fight by cancer's presence, means T cells have to make an absolutely ridiculous amount of new proteins like cytokines and molecules for attacking cancer. But the environment inside a tumor is rough. There's not enough oxygen, food scarce, tons of metabolic waste, so the T cells quality control team starts falling asleep on the job. The endoplasmic reticulum's overwhelmed, the proteasom's jammed, autophagy, the cell's cleanup crew can't keep up, it all piles up, and misfolded or broken proteins start to clog the cell. The T cell triggers this so-called Protectoric StressResponse, TexPSR, a last-ditch program that, ironically, saves the cell. By making it give up fighting, this is no small tweak on old models. It explains why even releasing the brakes using checkpoint inhibitor drugs doesn't always work. If the T cell's core cellular engine is fried, you can step on the gas all you want. Nothing's going to move. And something I thought was neat, NID connects TexPSR to real-world puzzles like why CAR-Ts, these genetically engineered T cells, often flop in solid tumors even though they work great in blood cancers. It's not just the cancer, it's this internal exhaustion from stress buildup. Now, you're probably wondering, so what? What does this mean for treatments, or for you, if you're following this as a patient or just someone curious about where cancer therapy is headed? Well, if researchers can figure out how to modulate, tune, or heal the T cell's stress response by boosting protein-folding helpers, activating cellular cleanup, or maybe combining these new strategies with the usual immunotherapy drugs, there's a chance to resurrect exhausted T cells and really help more patients respond. I have to stress, again, I'm just presenting this neutrally. The field is moving fast, but nothing here is medical advice or absolute truth. Slight digression here, but does anyone else remember those kids' cartoons with cells as little workers on assembly lines? Maybe that's just my brain, but when I read about proteasome jams and unfolded protein drama, I just, I don't know, I picture a tiny workforce in total havoc. Anyway, back to the research. There are real challenges, like how do you target these stress response pathways in the right cells without causing autoimmune issues elsewhere? And will it work in humans, not just models? It's early days. But If this holds up, if it's true, some in the field are saying Nobel territory. Of course, only time will tell. Academia's all about finding more pieces of the puzzle, not finishing it for good. So, what can we take away from this paper? Well, if you're invested in the future of cancer immunotherapy, TexPSR is a new contender for what's actually blocking cures. And thinking about stress inside immune cells might open the door to bigger, bolder breakthroughs for everyone else. It's just a reminder that solid research, open questions, and persistent curiosity, those are the engines that move healthcare forward. All right, that's all for today. I hope you found this breakdown useful, or at least thought-provoking. Remember, as always, these are insights and not absolute truths, just more food for thought as science keeps pushing boundaries. I'm Richard Price, and this has been In Depth with Academia. Until next time, stay curious.